Okay, Ra. Good morning. Okay, fine. Uh, Download that material. Okay, I just prepared. Uh, when it comes to this lesson, right? Transport in plants. Summarizing the summarizing till 11.4 itself takes one class because there are n number of terminology it will come, and I don't think whether you guys remember or not. But let us have a quick recap. Just a summary itself it takes a lot of time, but it's worth, right? It's worth that time, okay? Because the next half of the lesson will be very easy. Okay, so the next half is nothing but transpiration, right? So you guys requested me to take this transpiration 11.4. I'm not wrong. Yeah. So what are the yeah 11.4 is transpiration, uptake and transport of mineral nutrients. 11.5. 11.6 is phloem transport, flow from source to sink. So these three things needs to be understood. Means again you need to have a recap on the first half of the lesson, because it deals with the short distance transport of minerals and other water, at the same time other solute particles, at the same time long distance transport. And then we will try to understand how to put all the learned first half of the lesson in the transpiration, and then the uptake and transport of minerals, as well as transport in phloem, for that matter food. So we will be talking about water as well as food in the next half of the lesson. So most probably it would be next class, or in this class we will try to touch till transpiration. Okay, have you guys downloaded that material? Yeah. So I mentioned even number of the topic also. So it will not have any confusions. Okay. So let's start. Okay. Good morning, and we'll start with a very basic thing, that is just quick recap, of course. So we will be dealing in this chapter. We will be dealing with the transport, right? For that matter, how come this water and food particles are able to transport, or they are transporting from root to shoot, and from source of food material, nothing but leaves to different parts of the plant. So that is what we gonna learn in this chapter. That we already took this chapter, and you guys missed a few of the parts. So we'll be discussing about movement across shorter distances as well as longer distances. So which occurs through tissues, or for that matter, we have plant tissues like xylem phloem, or we have different cells as well, and through which how this movement of you know particles would takes place. Just to simply put forth, right? The smaller distances, the solute particles may travel with the uses of mechanism like diffusion, right? Facilitated diffusion or active transport, right? So on overall, what we're gonna learn is that transport over longer distances proceeds through vascular. For example, if the vascular system is involved, right, that would be involved in longer distances, transport of this minerals and food source, right? I mean food particles. and that is termed as translocation. So this translocation comes twice. So where once we we talk about when we talk about longer distances, transport of these materials, then we will understand better. So this longer distances through xylem and phloem, nothing but vascular system. And you should be remembering that this organic and mineral nutrients, they can transfer in multi-directional transport. It means it can go in a different directions as well. It's not, if you remember that, water flow in xylem is unidirectional. One comes to phloem, right? And one comes to food particles, right? It is of, two direction, bidirectional, because there is source is somewhere located, leaves or like that, and it needs to travel down to the different parts of the plant. So likewise, when it comes to, organic and mineral nutrients, they have multi-directional as well. So these directions, everything we will come to understand. Okay. So first thing, if you see in this material that I already gave, that is means of transport, right? It can occur through diffusion, facilitated diffusion, and active transport. So why I made these flowcharts is because it takes a lot of time and energy to recollect, especially the botany is concerned, and plant physiology is concerned, photosynthesis and this transport and respiration. These chapters are very important, and they will have a lot of terminology. So to avoid confusions, I made these flowcharts, and for time being, I gave different colors, right? If I have more time, I will spend time on having a specific colors as well. So meanwhile, you just go through this flowchart. First one is means of transport, that is diffusion, facilitated diffusion, and active transport. Right? Are you able to follow what I'm saying? Is it clear? My voice and all? Are you able to follow? Okay, fine. Okay. So we'll move forward. So we'll be discussing about not much discussion on this topic. Simply, I will try to explain these terms once again so that it will be clear for the next part of the lesson. Okay. So first one, what we will be discussing about diffusion, right? Diffusion is something is a passive kind of a activity, right? And we are trying to see that this means of transport within the cells or from cell to cell. So we'll see, because we are trying to understand. First one is means of transport in shorter distances, right? So how within the cell, right? Within the cell means cell organelles, right? Within the cell means from cytoplasm to cell organelles, and from cell to cell, what is it mean? So when it comes to diffusion, its movement by diffusion is nothing but it's a kind of a passive kind of a movement, right? It might take between cell to cell as well, but it will take over shorter distances. Okay, for that matter, intracellular spaces. For example, cell. There are two cells, right? So within this intracellular spaces, diffusions may occur. Okay. And if you see this simple diffusion, right, it doesn't require any energy. Okay, it doesn't require energy. Right, I always give one example, right? When we have a spray, right? When we have a spray, like it moves from higher concentration to regions to the lower concentrations. So with the just concentration gradient, right? These particles will move through this passive diffusion, which is a very slow process. That's why we call it as passive. Okay. So in the simple diffusion, what we observe is, without any involvement of energy expenditure, right? So this movement of molecules will take place, right? And this diffusion is very important process when it comes to exchange of gases, right, within the plant body, right? And if you see this diffusion rates, right, so this is a diffusion, simple diffusion, and how this diffusion can be affected. And this diffusion can be affected by concentration gradient, at the same time permeability of membrane, right? If there is a membrane which is actually separating two different cells, right? So based upon the layer in which these molecules are diffusing also matters. It means this diffusion rates are highly depending upon the concentration gradient, from higher concentration to lower concentration, permeability of the membrane, at the same time temperature, temperature, at the same time pressure. So these are the uh, four different factors that affects the rate of diffusion. So this is about simple diffusion, right? I think it's clear, right? So first we talked about means of transport. We are talking about the shorter distances transport of water. So means of transport, that is diffusion. That we talked about diffusion, which is a very which doesn't involve any energy expenditure, right? And then let's talk about the facilitated diffusion. So one by one we will try to finish as early as possible. I told now these terms, if you don't, they will be, they will, these all terms would look little similar when it comes to, these processes. So I'm going a little slow again to even for recap. It's really waste of time if we rush up things and if you don't revise properly. So let's have one more class. That's not an issue, right? Maybe next week we'll have. Okay, so let's understand the what do you say, the second process, that is facilitated diffusion. Okay, so when it comes to facilitated diffusion, what is actually facilitated diffusion? So in the name itself it is there, facilitated. It means someone or something is actually facilitating this diffusion. Now we know the simple diffusion, and but to undergo this facilitated diffusion, or we need to understand which molecules does actually goes, or which molecules that undergo this facilitated diffusion. So with that examples it would be more easy, right? So for example substances like hydrophilic in nature, right, water loving, right? So these substances may have a little difficult tendency to cross across the membranes. So at that point of time, what we require, or the what cell requires, is that proteins which actually takes up this hydrophilic moiety containing substances, and they diffuse them into the cell, or outside the cell, right? So this movement of this these molecules which are difficult to pass through the membranes with a simple diffusion would occur through this facilitated diffusion, or in which this facilitated diffusion is actually facilitated by proteins. And we know that if you look around your house itself, now you are sitting in the house or nearby your window, right? You might have seen that the window is something like a protein, right? So within which are embedded in the layers of the plasma membrane. So these proteins are outside the membrane. So these proteins, like transmembrane proteins are there, right, or surface proteins are there, right? So they facilitate this transport or movement of this molecules. So for that, so even though if it is a facilitated diffusion, we need to understand one thing is that for diffusion to occur, there needs to be a concentration gradient, be it simple diffusion or facilitated diffusion. Okay, but in facilitated diffusion, few molecules which are not able to transport or move, the movement is not possible by simple diffusion, though they have concentration gradient higher to lower. These proteins will be facilitating the diffusion of those kind of molecules. But even in facilitated diffusion also, we need concentration gradient. Is that clear? So when it comes to this facilitated diffusion, one more point we need to understand is that, sir, what is a moiety? Yeah, I told right, moiety is something which is a functional group. If you see, uh, acetic acid, or for that matter, any amino acids, right? Right, they will have a functional group moieties, in the sense like those are the uh, you know for example, I'll give one example that would be, so moiety is nothing but a like R group in any one of the organic molecules. If you see, so they will be involved in some they will have some this involved in chemical reactions as well. Okay, for that matter, what makes an acid acid, right? So what makes a lipid hydrophobic, for that matter, and what makes some molecules hydrophilic in nature, right? So which are water loving, so they are reacting with this water molecules. If you are not, they are hydrophobic, they don't react. So those are the specific group of atoms, right, within a molecule. Is that clear? So they are specific group of atoms within a molecule. So fewer water loving moieties will be there, fewer water hating, like water phobic, hydrophobic also will be there. Yeah. So like that, this moieties will be there in different molecules, even proteins, etc., where they actually react with the surroundings. So the hydrophilic moieties, which the substances which has this hydrophilic moieties, needs to be facilitated by proteins for their diffusion. But for the facilitated diffusion, again, what we have to keep in mind, we have to keep in mind that though it is facilitated by proteins, it has to have a higher concentration gradient, higher concentration to lower concentration. And one more point to be added here, that is, it doesn't involve any energy consumption. Energy expenditure is not involved in facilitated diffusion. Means here energy in the terms of what? Cellular currency. Energy in the terms of what? What I'm talking about? Energy in the in terms of energy in terms of what, guys? I'm telling that this facilitated diffusion doesn't require any energy, right? What is the cellular energy? Cellular currency. Yeah. Hmm. So there is a lag in the video, I guess. ATP, adenosine triphosphate. We know how the energy is generated, right, by this ATP, right, or how energy is stored in the form of ATP, right? Yeah. Here. Very good. We are in the first slide itself. I think you guys asked for summary. I told now it takes a lot of time. this okay, or shall I just rush up? Okay, this active transport is there, this diffusion is there, this water potential is there, pressure potential is there. If I tell like this, within 5 minutes I can finish it off. What you require? It's up to you. You need explanation, or you need just you wanted to see the contents. Contents you can refer in your textbook as well. Okay. Okay, fine. No, I will make it a little fast. No, I understand the importance of this terminology. That's why I'm going a little slow. But let's see if we can go a little more than 11 o'clock. That's if that is okay, we will move forward. Okay, next diffusion. First slide is going to going to. I mean, it's getting over. So when it comes to facilitated diffusion, right? So it can facilitate in two means, right? So there are in this facilitated diffusion, right? Because I already gave during the class, right? I gave an example of direction, right? I think I missed one of you know passive imports and deports. Okay. Yeah. So I gave one of the during class itself. I gave a few examples that how these molecules would facilitate their diffusion, or what are their direction, right? There would be a direction for facilitated diffusion, like uniport, antiport, symport. Okay, so passive symports and antiports. If you see this, in this we talk about the direction of this movement of the molecules. So uniport is something. If a substance A, molecule A would move from one direction to the other direction, either inside to outside, outside to inside, right? And when it comes to antiport, so there will be two molecules, one A and B, right? So one would move in this direction, maybe from outside to inside, and other molecule would come from inside to outside. So this is antiport. So this is a protein which is embedded in the transmembrane protein like this, right? If this is a cell membrane, my watch is like a protein, right? Right. So through this protein, right? So it is moving like this. Either the protein would allow in unidirectional or antiport. Uniport single direction, antiport is opposite direction, or symport is two molecules can move at a time in one direction. So these are the three directions that actually what do you say, this flow is correct, sir. Sir, no need to rush. Okay, fine. So in this facilitated diffusion, we have nothing but direction of the movement of the molecules as well. Here, yeah. And one more thing is that in this facilitated diffusion, even we will come across this porins, right? Porins are nothing but these are the proteins that form huge pores in the outer membranes of the plasmids, plastids, or mitochondria. Okay, so these porins, right? They facilitate the facilitated diffusion. So you may get as in NEET also. You should be remembering that what are the functions of the porins. Where does this porins exist? So if you see this porins exists in plasmids and mitochondria, and even in some bacteria also you will find these porins, right? And we call them as if these porins right, they're actually transporting the water, right? So they are if they are involved in the water transport or movement of water or acting as water channels, we call them as aqua porins, right? So they will be even this these porins, one of the important function is that they can even pass small proteins. Usually proteins would not actually travel through the plasma membrane, but through these porins, right? Through these porins, even small size, small size proteins also can travel. Okay, so this is about the facilitated diffusion in which there is no an energy expenditure. Okay, fine. So let's move on. Next, we'll go to the active transport. So when it comes to active transport, what is the difference between active transport and passive transport? So in passive transport, we don't require any sort of energy, be it diffusion, simple diffusion, or facilitated diffusion. Facilitated diffusion involves proteins, porins, aquaporins, right? Though they involve proteins to transfer these molecules, they need concentration gradient higher to lower. And this diffusion and facilitated diffusion, which are passive transport, doesn't require any energy consumption, right? And when it comes to active transport, it requires energy expenditure. So that is the one of the major difference in the active transport, right? So active transport is nothing but, right, active transport is nothing but a transport of or movement of molecules from, right, from one concentration to other concentration. Now here through membranous protein pumps, these molecules are moving from one concentration to other concentration. And why it is needing energy is that these molecules are moving against the concentration gradient, maybe from lower to higher. So it needs some sort of energy to really take up these molecules and put it in this higher concentration. Okay, so this active transport involves energy consumption, and it uses this energy to pump the molecules from lower concentration to higher concentration. It means against the concentration gradient. Okay, is that clear? So other this this movement of molecules from lower concentration to higher concentration, right? It is otherwise called as uphill. Okay, so with this, first slide is done. I hope you guys are very clear in this, and I'm telling again and again for the NEET +1, the 11 portions are very important, especially the physiology part, plant physiology. They are as good as organic chemistry or any derivations you derive in physics, because they are very volatile and you have to keep practicing, just like math, genetics, and this physiology. No other shortcut. Though I am giving in a simple terms, the flowcharts, etc., would definitely help you during the revision, but you can't just rely on them. You will be at most confused at the end. So I really suggest you to use this time if you are free and if you are having no much burden on you during this COVID lockdown, to utilize at least an hour, 1 hour. I don't say that you have to read for 18 hours in this lockdown situation, because it is already a distressing situation that we are locked down in a four walls. We are not seeing anyone other than our family, or sometimes family members may get sick. So it is very tiring experience that anyone can have, and you guys in young generation are experiencing that. You guys are able to give a future direction for your next generations. So you can say that, okay, in 2020 we have seen a pandemic, like how like we have seen even I didn't see any pandemic in my lifetime. Maybe by that time Edward Jenner, these days, are already took care of that, smallpox, etc. But what I'm saying is, you witnessed a very it's 2 minutes break. Okay. maybe not 2 minutes, maybe 30-40 seconds. Okay, so this situation is not that easy to take up, and even there would be some sort of illness as well, right? It would be a little depressing or sometimes happy, of course. uh, but I don't want to put any pressure at this juncture, right? Take your time, keep proper relaxation time as well. Talk to your parents or friends, whoever, non-judgmental, right? And try to read at least this 11th standard portions for 1 hour or 2 hours, right? And already your school schedule is going on. Maybe if the school schedule is stopped, you please spend time on this plant physiology and animal physiology. Okay, fine. Let's go to the next slide. That is, I gave in the next slide. I already gave the differences between the simple diffusion, facilitated transport, or diffusion or active transport. Okay, I gave the properties, which is just a NCRT text extracted, table extracted from NCRT text, right? Source I will mention here. So what are the properties that you don't see in simple diffusion and which you will see in facilitated diffusion and which you will see in active transport is very clearly given once you go through. For example, requires ATP, simple diffusion doesn't require, facilitated doesn't require. Uphill transport can be taken by simple diffusion, at the same time facilitated transport. And you have highly selective in case of simple diffusion, no. Uphill transport, no. Right, when it comes to facilitated transport also, uphill transport, no. So likewise, I have given one table which is given in NCRT textbook as well. Once you go through this after the class. Next, let us go to the next one, that is 11.2, that is plant water relationship. So this is more confusing concept you can ever get in your 11th standard textbook, especially in botany. Okay. So these terminologies are require little in-depth understanding. Otherwise, you will be lost. Okay, let's move into this. Yeah. So when we talk about plant-water relationships, right? Why exactly we are talking about water here? We know that plants do not survive when there is no water. Or water is very essential for all physiological activities of any living organism, especially the plant, right? Which in which it provides, it acts as a medium, right? Why water is so much important for plant, for that matter, right? Is just because it acts as a medium to actually medium for what, for all this process to occur. For that matter, you need this medium, water as a medium, to actually facilitate this diffusions or transport or movement of minerals or food particles, you name it, right? So within the protoplasm you have a higher water content within the cells, right? So the protoplasm is nothing but actually it is comprising of the water. So that's what water is very important, and we will be learning this. Uh, how come we can really relate this water with the plant activities? That is what the entire 11.2 is talking about, right? Yeah. So let's move into the relationships. So we'll be talking about transpiration in the 11.4 session, because water which is used up by the plant, at the same time the excess water is sent out of the plant through transpiration, right? Through the process of evaporation through leaves, that is nothing but transpiration. So this transpiration we'll be talking in the next junction, right? Especially terrestrial plants. So when you see this relationship what are the phenomena that this water has in these plants, that let's start one by one. So when you see, I gave flowchart also, that is plant water relationships can be understand by water potential, osmosis, plasmolysis, and imbibition. Imbibition, still I remember, I took this, when I took this topic, even it, it feels that I took this imbibition topic just yesterday. So that is impact I have in my mind when I take this imbibition, which is a special type of you know one of the special type of the process. We will talk about this imbibition as well. So let's right, when we talk about this imbibition, right, at the end we will talk about this. So it is special type of diffusion. So it's has a huge impact in my mind. So let's start with the water potential. So water potential, what is water potential? So within the water potential we can understand two different things, that is one is solute potential and pressure potential. So water potential, as there was a small formula for the water potential as well. So if you see that a relationship of water potential, you will get to know. So water potential of a cell can be affected by both solute and pressure potentials. That's what. So the psi W equal to psi S plus psi P. So S is solute potential and P is pressure potential. So you have to recollect the terms, what exactly the water potential means. So water potential is nothing but it's a fundamental, it's a movement of water, right? Movement of water within the plant. So how exactly this water movement can be understood with the help of this small equation, that is W equal to S plus P, means psi W equal to psi S plus psi P. So this solute potential and pressure potential are two main important components of this water potential to occur, right? So water potential you would have studied in physics even, right? We have studied about physics. Any object has its kinetic energy, right? So when it comes to this water molecules, they do possess kinetic energy, right? So they tend to move. So they tend to move. So when the are you guys there? Am I clear? Am I clear, guys? So we are talking about this water potential. So when we talk about water potential, greater the concentration of water in the system. For that matter, we'll be talking about purity also here. So when we talk about the concentration of the water within the system, it will have more kinetic energy. So it has more water potential, as simple as that. So within the water potential system, you have a solute concentration, at the same time pressure potential, right? If you yeah. For example, you consider uh, okay, if you consider this as water potential, within which you have solutes, right? So solute potential, right? Solutes, if more solutes are there, these relationships I already gave in the class as well. So if this solute potential is more, how it is affecting the water potential, right? So that I already gave, we will discuss little later, right? And it has got some pressure potential. If we apply some pressure, right, what happens to the water potential. That's what we are going to discuss. Is that clear? Is everyone is there? One left, it seems. Who are there actually? Srividya there, Kritika, Mrita. Yeah, someone left in the middle. Is it Srividya, Amrita? Three members are there. Yes. Yeah, someone joined again. Okay, please don't leave in between. Okay, I can see everything here. Actually, who is there, who is not there. Fine. So let's move on. Just a minute. So I hope you have running notes for this. I think whoever missed the class, that's what they are taking, right? Right. So I already told in during the class that even now I've shown that if you take water in the beaker and you put some solute, so it affects the water potential. So more if the water concentration is more, it means it has more water potential. As simple as that. It means the solute potential should be low in that system. As simple as that. Fine. Let's move on. So if the solute potential is increasing, what happens is, it, the purity of the water comes down, right? It means that water concentration is very low, so that the water potential will be also low. As simple as that. So these are the relationships that we can see within the water potential. How this solute potential affects the water potential, and if we apply some pressure potential to this system, right? To this system, how does that affect? That is what it is given in this this entire topic. For example, if a pressure greater than atmospheric pressure is applied to a pure water or a solution, for example, more than the normal pressure, right? If if the pressure potential is applied, if a pressure is applied to that cell or within the protoplasm, right? What happens is the cell become turgid. For that matter, what happens is the water potential would be increasing. So pressure the greater than the atmospheric pressure. Atmospheric pressure already it would be existing, right? So more than the atmospheric pressure, if a pressure applied for a pure water or a solution, what happens is water potential increases. So solute potential and water potential are inversely proportional, and this water potential and pressure potential are directly proportional. As simple as it. So if you see, different types of concentration gradients, right? We have seen that there is a flaccid cell, there is an isotonic solution, and there is a turgid cell, right? So when the pressure potential is high within the protoplasm, within the cell, what happens is the cell becomes turgid. So that's how this water potential, the components of water potential does really affect the cell. Is that clear, guys? This water potential topic. If you once you go through, it will be more easy. Once you go through the, what do you say, class notes, and once you go through the NCERT, it will be more clear. Is a clear water potential, pressure potential, how water potential increases and decreases, and how this solute potential may affect the water potential, and how this pressure potential may affect the water potential. No doubt, right? Yeah. So one important point we need to understand is that water will move from always the system which containing higher water potential to the one which is having lower water potential. As simple as it. So we are discussing about the movement of the molecules within the shorter distances, right? And we are trying to understand the relationship of this water with the plant or cells, within the cells, right? How this water is interacting, what is the phenomena. So that's what we are discussing within this. And the next one is osmosis. So I hope this water potential concept is understood better. Okay, so simple equation we can remember is this: W equal to S plus P. Okay, when we add up, sum up this solute plus this pressure potential, that gives the water potential. Okay, now let's move to the osmosis. So osmosis, again it undergoes with a pressure gradient or concentration gradient. Two aspects we will trying to discuss. And this osmosis, is, if you see, this osmosis is it is specifically actually occur within or if you say this, the diffusion of water molecules across semi-permeable membrane. So diffusion is something like a gaseous molecules, right? When we talk about water molecule diffusion, right, through the semi-permeable membrane or differentially permeable membrane or differentially permeable membrane, the water molecules move. That movement is called osmosis. Okay, and this movement, the net movement of this entire water molecules would be dependent upon two aspects that I already gave in the flow chart, right? That is pressure gradient and concentration gradient, right? One is pressure gradient and concentration gradient. So this osmosis is actually dependent upon the pressure and concentration gradient. So if the pressure is changing, how does that affect the osmosis? If the concentration is changing, how does that affect the osmosis, is what we learned previously. So within this, osmosis is little easy topic, right? Because we have a simple experiment or demonstration of osmosis is already given, where when when you're applying some pressure right? In which we'll take a funnel and we will keep what is this a semi-permeable membrane, and we put it in the water, right? And we will leave it to it. And the movement of this, the rays of this sucrose solution could be noted down. But when we apply the same pressure, how this really differs, that is given in a simple demonstration. So in that demonstration, we'll come across the pressure, right? Pressure gradient. How much, if the pressure is increasing, how does that affect the osmosis? If at all the concentration is increasing or decreasing, how does that affect the osmosis? Is clearly mentioned, and I think it doesn't require much discussion on this. Okay, the simple demonstration, if you try to go through, and if you don't understand, let me know. Okay, so if you see, always the water will move from a region of higher chemical potential, right? Nothing but here potential in the terms of, we are using in the terms of concentration. So higher chemical concentration to the lower chemical potential. So water will move from higher chemical potential to the lower chemical potential. So these water potential and these two needs to be combined. You need to overlap these two. Then only you will you will understand better, right? What we are talking here is chemical potential, right? Or concentration. So it moves from the regions of higher concentrations to lower concentrations until it acquires equilibrium. Once the equilibrium occurred between the two systems A and B, what happens is there won't be any further movement of water molecules. As simple as that. Okay. Clear? Okay, fine. So this is about the osmosis. Very once go through the that what do you say, demonstration, then it will be very clear. And coming to the plasmolysis, plasmolysis is again it's kind of a demonstration. Also we can see it's a very simple process, right? How does this plant cells behave, right? Or the cells within the tissues behave, right? With regard or in terms of water movement, right? So this would be depending upon the surroundings. If this is a cell, right? If this is a cell, so if this is a cell, right, the diffusion of water molecules would depend upon the surroundings. Nothing but we talk about in the terms of concentration. And here three types we can observe. That is, we can be understanding this plasmolysis based upon isotonic, hypotonic, hypotonic. So isotonic solution is something, right? The cell is there within the same concentration, right? So there won't be any transfer of water molecules, right? There won't be any movement of water molecules from the surroundings to inside, from inside to outside. So that makes it iso, similar conditions. So the external solution and inside the protoplasmic solution has a more or less the same concentration. So that chemical potential. So that what happens is that there won't be any much, or there won't be zero movement of the water molecules. That makes it isotonic. And the isotonic solution, if you keep a cell in an isotonic solution, right, they appear to be flaccid. Okay. And when it comes to hypotonic, in the term hypo means less tonic concentration, less concentration, it makes it turgid. Okay. It means when does this hypotonic situation occurs? So hypotonic situation, the process of plasmolysis is usually reversible, of course. It means that it can be, for example, you kept a cell in an isotonic solution, and then that cell can be kept in an hypertonic solution, and it behaves in a different manner. and you can keep that cell again back into the hypotonic solution, so it behaves in a certain fashion, right? So it kind of a reversible kind of a process, plasmolysis. So based upon the available concentrations in the surroundings and within the cell chemical potential, this cell would behave in such a manner. So when you place a cells in hypotonic solution, it means higher water potential or dilute solution, right? So when compared to cytoplasm, so cytoplasm is more dense than the the outside external solution. So water diffuses into the cell. So it causes cytoplasm to build up some pressure, right? Are you able to understand? So in the cytoplasm, so we talked about solute potential, right? So the solute potential is too much within the what is a cytoplasm. So there are many solute. You got very it's very interesting, guys. If you spend some time in this topic itself, okay, in this topic, water potential topic itself, you spend a day, no problem. But it is very beautiful concept. You will regret if you don't if you skip this topic. If you will regret. Okay, it's that beautiful concept. So if you take a cell, right, the cell contains protoplasm, cytoplasm, and it is made up of n number of molecules, right, solute particles. So that makes it more solute potential, and it has less water potential. So always you remember the water potential relationship or how it behaves. It always water tends to move from higher water potential to the lower water potential. What makes lower water potential? Higher solute concentration or higher solute potential. That's what inversely proportional. Once the solute potential is too much in the cell and the external solution has less or diluted solutes or more or higher water potential, so this water would tend to move inside the cell. Once the water is moving inside the cell, what happens? It exerts pressure inside the protoplasm. So this protoplasm exerts pressure in all the directions into the cell wall, and that makes looks bulge, turgid. So this is what we say turgid pressure, turgid. It makes it turgor, right? That exerts the turgor pressure which builds around the cell wall. For example, the water is entering into the my room and it is exerting pressure onto the walls and the room is bulged. So that makes it when the cell is in hypotonic solution, turgor pressure operates. Why? Because now you are able to relate, guys. Are you able to relate what I'm talking about? How important the water potential concept is? Are you able to understand? So within the cell, right, there are lot of solutes are there. So outside there were no much solutes. So it has more water potential because water concentration is high, right? Water potential kinetic energy is high. So the water tends to move from outside diluted solution to the inside concentrated solution because it has got more water, more solute potential. So when the water moves inside the cell when you place the cell in hypotonic solution, right? So water, this protoplasm which is present inside the cytoplasm exerts turgor pressure on the cell wall and it looks bulged. So one is isotonic, the movement of water molecules it came into an equilibrium, and because external and internal concentrations are the same, so there won't be any movement of the water molecules. Second one is hypotonic solution. During this hypotonic solution, the outside external concentration is diluted or less solute particles are there and more water potential is there. So the water tends to move inside the cell which has got more solute potential or solute particles and less water potential. You have to keep writing all these things. Then only I already gave it in class as well. Okay, that makes this cell turgid. Okay, first one is flaccid. in isotonic solution, and this is hypotonic solution, turgid. Okay, and then finally we'll go to the next one is hypotonic solution. Hypotonic solution is something which has more concentration outside the environment, outside the cell, right? It has got more, more solute particles, so that makes the concentration hypertonic, right? Hyper means more concentration outside the cell. Inside the cell compared to the cytoplasmic concentration, outside concentration is more, so inside there are more water molecules than the outside, so more water potential inside and less water potential outside, and water tends to move from inside to out. So what happens? The cell would shrink, so it loses all the water within the cell. For example, it's like dries out for that matter, so all the water potential would be moved, all the water potential which is present, higher water potential which is present inside the cell, cytoplasm or protoplasm, would tend to, the water tend to move outside the cell, from within the from within the cell to outside the cell, because higher solute potential is there outside and less water potential. So always water moves from higher water potential to the lower water potential, right? That's it. The cell get plasmolyzed. Is that clear? Is that clear, guys? Remaining? Saima? Oh. Okay, Ra. So it's a very interesting topic. It's really hats off to the scientists who actually worked on this. So flaccid means no change, sir. Yeah, yes, isotonic. Okay. So you have three types of cells. One is plasmolyzed cell, and one is uh, turgid cell, and one more is flaccid cell. Okay. Flaccid cell is something which is an effect comes when the cell is placed in isotonic solution, and plasmolyzed cell is that when the cell is placed in hypertonic solution, and turgid cell, this is a phenomenon in which the cell is placed in hypotonic solution. Is that clear, guys? I will update this you know mm, flow chart with some other pictures that would be little interesting. Okay, so imbibition, let me take this imbibition and let us stop with the imbibition. Is that clear? Yeah, okay, fine, okay, fine. So let's move into the next end of the topic of the 11.2, that is imbibition. Imbibition is nothing but a special type of diffusion. Okay. So it is in the name itself, it is there, imbibe, right? It is something which is actually uh, taken up by some solids, right? Some solids, right? This water is absorbed by these solids is nothing but the imbibition. So that what happens is when they imbibe this water, right, they enormously or they rapidly increase their volume. Okay. For example, you have flax seeds, or if you take this basil seeds, right? If you see this basil seeds, you put it in water, right, and you see for 2, 3, or 5 minutes, you can see that imbibition would occur. So eventually, this is a solid particle, right? The seeds are made up of more of a solutes and solid particle, which rapidly absorb these water molecules and imbibe these water molecules, right? And they actually swallow. So this is one of the good example I'll try to show right now. Yeah. So if you can see these guys, right? So you can see these guys. I think everyone knew about this, right? So these are all seeds, right? And then you put it in glass. I really hate these guys. For health purpose, my I had to do this. Okay, I put some water in this, and you just try to observe. Are you able to see? You can see that they actually imbibe the water. I mean, imbibe the water. It means they take up this entire water. If it is a transparent thing, I would have shown. You can see, right? Are you able to see they are bulging? Yeah. I really hate these guys. People say that they taste very good, but yuck, I don't like. Okay. Still, you can wait for an hour. You can see more imbibition. As simple as it. So what's happening here? There is a higher water potential, right? So I pour the water. The seed has more solute concentration and less water, right? So less water potential. So now what happens within this cup? If you take, this is a system, right? Within this cup, water is there, seeds are there. So water itself is having more water potential, kinetic energy. So they tend to move inside this seeds. That is any case of how this water potential works. Higher water potential to the lower water potential. So what making lower water potential is that the seeds having low water potential. So this seeds tends to imbibe. When it comes to this imbibition, is a special type of diffusion in which this water is able to accumulate, or water is absorbed within the seeds and they bulge over time. A simple. Seven, I don't want to drink that as well. So, okay. So imbibition we refer to a diffusion because water is moving along the concentration gradient, as simple as this. Okay. Why? Because this wood or other seeds, they doesn't have higher water potential or higher water molecules, so they tend to absorb this water from the external environment to inside the seeds or woods. That's what makes in this imbibition. In addition to this imbibition, that is, for any substance to imbibe any liquid, affinity between absorbent and the liquid is also prerequisite. But for that matter, to imbibe, right? The solute particles, it cannot, this plastic cannot imbibe anything. Though it is a solute, it cannot imbibe. It means the affinity between this absorbent and the liquid is also prerequisite, in the sense like it is very much essential. Any solute cannot really absorb the water as simple as just like that. So even absorbent chemical nature is also, solute nature is also important for the imbibition to occur. Is it clear? uh, I think I wasted water. I'll try to drink. Yeah, I don't know how these people will drink this, but this, it's healthy actually. It's cool down the system. Okay. Okay, Ra. You just go through this water plant today completely. Half a day or at least 2 hours you spend time on this water relationship. You will really love this subject botany when you really sit and try to to relate. It's quite a difficult thing, but see, right now I did something which is there in the home itself. That's how you have to have an open mind to really see. And especially this, when it comes to botany or bio or any subject, it's also depends upon the teacher, that how they teach as well. Sometimes it may reach, sometimes may not reach. So please, try to spend much time on this water potential concept, so that you will really love botany as well. Trust me, because I love botany, especially this concept. Okay, Ram. So next class we will be discussing about so 11. So 11.3, simple plant absorb water, apoplastic, symplastic pathways. Once you go through today, and it will be very easy, right? For that matter, absorption of water through roots. How does it happen? So from the root head till the xylem, vascular tissue, how this water is actually passing. I gave it in material, very clear images I already gave, and you have to remember that in NCERT they given wrong diagram, picture representation. For apoplast they gave symplast, from symplast they gave apoplast. Okay, in the class I will mention again. So what I gave in this diagram in the material now, right? You try to follow that. Is that clear? So okay, we'll stop here. We'll continue this class again. Okay. So once you go through the symplast and apoplast, okay, then have a nice weekend, of course. Hope everyone is healthy. Saima, you're okay? Your health is fine now? Yeah, okay, fine. Okay, take care, guys. Whoever there now, 5 members, please take care of you, and please go through this contents again. You throw the book aside, you will not get anything. Okay. Okay, then fine. See you in the next class. Meanwhile, you just take care of yourself. Bye. Thank you, guys.